Hey everyone, Yamina here. For those of you who didn't know, I spent 5 years at Rockefeller University in Tom Sakmar's lab. I did have a blast there. Before letting you listen to the regular introduction and the podcast episode, I just want to express my gratitude to Tom for allowing me to work in his team, and say thank you to the Sakmar lab for sponsoring episode 50 of the Dr. GPCR podcast. I'm excited to share this episode with you. Can you believe it? We've reached episode 50. Alright, I'm going to let you go. Listen in to the episode. I hope you enjoy it. Again, thank you, Tom, and thank you to the Sakmar Lab for sponsoring this episode.
Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. We are working on a new program geared towards transitioning from academia to industry. Powered by Dr. GPCR, our new program is called Life Career Job. The goal is to support you in your efforts to find your next job in industry, plan out your career trajectory, create a life, and support the lifestyle that works for you. If you'd like to be notified when this program becomes available, fill out our short survey at lifecareerjob.com. This new ecosystem will also include a new podcast that will be launching in September 2021. Our guests will be scientists, recruiters, and hiring managers, and everything in between. We want to help PhDs find their ideal career path and inspire you with real-life stories. The second edition of the Dr. GPCR Summit will be held from September 13 to 19, 2021. We are planning a combination of live talks, pre-recorded talks, and live workshops. We have a great lineup of live talk speakers, including Dr. Brian Roth, Dr. Patrick Sexton, and Dr. Jürgen Weiss. Also, we are happy to announce that we'll have a live Q&A session with Dr. Bob Lefkowitz and Dr. Randy Hall to talk about GPCRs, science, and Dr. Lefkowitz's memoir. Visit drgpcr.com/summit today to see the full program. We're also launching our GPCR consulting services. You'll soon be able to find the profiles of our carefully selected consultants on our website at drgpcr.com/consulting. You'll be able to find the help you need for your company. Stay tuned. Are you subscribed to our YouTube channel? If not, please subscribe today. It's not only a great way to catch up on our recorded events, such as the Dr. GPCR Virtual Cafe, but it's also a great way to support Dr. GPCR. And now, let's dive into this episode.
Hello, everyone. This is Yamina from Dr. GPCR. Today, I have the pleasure of having with me Dr. Tom Sakmar. As many of you know, Tom is one of my former mentors from the Rockefeller University. I had the best 5 years of my life in New York. Hi, Tom.
Hi, Yamina. It's good to see you again.
Great to see you again. I think this is our once a year chat, and this time it is recorded. Since I've left the lab every year around the same period, I think May or June, we get together and chat. And today is at the end of last day of April, and I'm glad that we're doing this on the record.
Well, it's always good to be on the record, although I do miss the informal off the record chats as well.
Definitely. So can you tell the audience a little bit about yourself? And I'm sure a lot of people know you, but just for those young scientists who's never heard of you, like I hadn't heard of you before I Googled you and sent you an email to ask you if I could join your lab.
Well, that's interesting. I was, my lab existed before Google, so not everything you might think is on Google that should be there. But here we are in New York City. I'm sitting in our lab conference room at Rockefeller University, where I've been a faculty member since August of 1990, so 30 years at Rockefeller. Our lab is interested in, and has always been interested in, transmembrane signaling. We are what I'd call now more of a chemical biology lab than anything else, I guess. Our animal budget is 0 this year, which tells you a little bit about our focus. In my own career, I started out working on GPCRs before I knew I was working on GPCRs, because I was a postdoc in the 80s in Goban Khorana's lab at MIT in the chemistry department. And I was working on the visual pigment Rhodopsin. Rhodopsin had just literally been cloned, the bovine Rhodopsin, and then human Rhodopsin had literally just been cloned as I joined the lab. Our lab was one of the labs that cloned the first G protein alpha, beta, and gamma subunits, in this case transducin from the retinal rod outer segment. And my mission was to express these cDNAs heterologously in mammalian cells, which was no mean feat in 1986. And that worked out. I had a great group of colleagues that I'm still close to, even 35 years later. Dan Oprian and Sriram Subramaniam, Roland Franke, people like that, Sadhu Karnik. These folks, we had a team effort, but we all had our individual projects. I was lucky that Goban Karana gave us a certain amount of freedom, day-to-day freedom. He wasn't a micromanager, so we could pretty much run with anything we wanted, although he also, of course, was a chemist, and so we were always interested in pushing the technologies in the molecular biology realm. At that time, one of his former students, Mike Smith, had just invented this mismatch primer-based mutagenesis with M13 as the phage that was used to do the site-directed mutagenesis, where you could make single missense mutations in your gene of interest. And Goban was really excited by this strategy, so we worked out that method in parallel with gene synthesis for our genes of interest. So we were doing sort of competing strategies to make the first site-directed mutants of rhodopsin and G proteins at that time. And I think that we did make probably the first mutants of the GPCR. There may have been other, of course, there were others out there who were making mutants, Bob Lefkowitz group, others, the Richard Dixon, Kathy Strader groups at Merck were doing this. A few other groups, of course, were interested in making mutants. But one advantage we had with Goban's background was that we started making synthetic genes for the proteins that we're trying to express in HEK cells. In fact, we started expressing things in COS cells. But making synthetic genes allowed us to do mutagenesis by restriction fragment replacement, because if you make a synthetic gene for your kilobase typical GPCR of interest, you could introduce unique restriction sites such that the plasmid would have a limited number of sites, and you could, because of the degeneracy of the genetic code, introduce or remove sites within the gene of interest. So if you wanted to make a mutant, you could just pop out XHO1, BAMH1 fragment, for example, make a 50 base pair synthetic duplex with your desired mutation, and then pop it back in to the gene.
Which sounds, if you allow me to interrupt, which sounds really trivial now that you're talking about it, but it was not, because remember, it was 1986.
Yeah,
and
it was 1986. We had to make our own ligase enzyme, which one of us would do every couple of years and freeze down a bunch of tubes, keep those in the freezer. Things weren't kittable as they are now. There weren't kits for everything. But I don't want to totally date myself and talk about what I was doing 30 years ago, but I have been working on GPCRs since 1985, which I think is pretty impressive.
I think so too. Let's go back just a little bit in time so that we can come back to the present and to the future. So you're trained as a physician, and then you ended up doing a postdoc at MIT. How did that happen? What was the element that caused you to get interested in lab work and in working at MIT?
Yeah, well, there's a story behind that. I can tell you, give you the highlights.
Of course.
I was a chemistry major as an undergrad at University of Chicago, although I must say I wasn't a hardcore chemistry student. I ended up with a BA in chemistry, which sounds kind of weird, but I took a lot of humanities courses. I took 4 years of French, but I also loved being in the lab, and I, so spent time on internships and part-time jobs in the lab trying to raise money to pay for things. And then I had this interesting opportunity where I applied for jobs, for summer jobs in 1976. And I ended up getting a job offer. In those days, you used to get job offers in the mail. A letter arrived from the Food and Drug Administration said I've been accepted as an intern for the summer. And so I went there and ended up working at a place called the Bureau of Biologics on the NIH campus, which is now the Center for Drug Evaluation. Mm-hmm. It's actually a research lab based agency within the FDA. And that was a fantastic experience. I worked with a guy called Darrell Liu, and my section chief was John Robbins. John Robbins went on to win a Lasker Award. Darrell Liu had a very distinguished career in protein biochemistry. And at the end of that summer, I was at University of Chicago as an undergrad. At the end of that summer, Darrell Liu said, you know what, I have an old friend at Chicago Biochemistry named Bob Heinrichson. And we were postdocs together at Rockefeller University. In the 60s, we worked with Stein and Moore, the famous chemists who invented amino acid analysis. Anyway, Bob's my best friend. I'm going to write him a note, and you can go talk to him and see if he'll let you work in his lab next summer or whenever. So I got back to Chicago after that summer. I had a great time in Darrell's lab, and I made lifelong friends there. And by the way, I was working on trying to isolate a membrane protein there in 1976. So Bob Heinrichson, of course, with that letter of introduction from Darrell Liu said, yeah, sure, you can work in the lab. And so from then on, basically from 1978 onward, I had a bench in Bob Heinrichson's lab in the biochemistry department at University of Chicago. And in those days, his lab was doing amino acid sequencing with the old spinning cup sequencing machines. It was before solid phase stuff. And so my job was to purify phospholipase A2 from snake venom and work on the team that was trying to do the amino acid sequence of that. So because of that project, I got to work with, in addition to Bob, who's a fantastic scientist, I got to work with this famous peptide chemist called Tom Kaiser, who also ended up at Rockefeller. And I got to work with a structural biologist called Paul Sigler. So using the material I purified, Paul Sigler eventually crystallized phospholipase A2, and I got to know Paul Sigler and get interested in structural biology, but it was all related to membranes because phospholipase A2 has this so-called interfacial recognition peptide that allows it to bind to membranes to hydrolyze the phospholipids. Meanwhile, I was on a track to go to med school, so I just stayed at Chicago, went to med school, had a bench in Bob's lab the whole time, and turns out not only did I love being in the lab, but I love being in the hospital. So I was sort of
torn, torn between two.
Well, torn. I didn't feel torn because at Chicago, the hospital was right across the street from the lab. So I would be in the hospital, I'd go to the lab, I'd go home, I'd go to the lab, I'd go to the hospital. And so for those 4 years, I tried to do some work, but I also had my med school responsibilities. But the problem was that all the time I was in med school, I felt like I was, and this was 78 to 82, I was following peripherally the revolution in molecular biology. I was pretty well trained in protein biochemistry, but I was totally missing out on molecular biology, cloning, the whole DNA revolution. So I said, wow, when I finish med school, I obviously want to do a little bit of clinical training to get credentialed and get a license. But I also want to do some kind of a postdoc so I can learn molecular biology. I was committed to research. And then when med school finishes, you sort of choose a subspecialty. You don't know where you're going to be because there's this match system where you apply to places, you rank them, they rank you, and then on a certain day in March, everyone finds out where they're going to be. And I got lucky somehow and ended up matching at Mass General Hospital in Boston for my medical residency. So in 1982, I moved to Boston. I was an intern at Mass General, but it was a very academic group, that class of 15 people at that time, a lot of future luminary scientists in my class. And everyone was going to do some research type postdoc, almost everyone. So I immediately looked around Boston with the help of my mentor from Chicago, Bob Heinrichsen, and picked out a bunch of labs where I could contribute something in terms of membrane stuff, but also learn molecular biology. And I interviewed at 5 or 6 places. I won't say who. I was able to get accepted at every one for a postdoc, a sort of a traditional research postdoc after my 3 years of medical training. My original plan was to finish my medical training, do the postdoc, and then come back to the hospital to do subspecialty training. And my medical department chair liked that idea, John Potts. He said, yeah, fine, come back, we'll hold space for you. I never went back to the hospital. So, things started going well in the lab. I ended up choosing Gobin's lab. It was kind of interesting. Everyone I talked to said, how long do you want to do the postdoc? And I said, probably 2 years would be optimal. And Gobin said, how long do you want to do the postdoc? I said, 2 years would be optimal. He said, there's no way that 2 years is optimal. And he's the only one that really was honest. He said, you can't learn enough in 2 years. You'll just start learning what you have to learn, and then you won't be able to accomplish anything. I would say you should keep it open-ended, the open-ended postdoc philosophy. But that had some influence on me. And he said, yeah, don't worry, I'll support you and you'll get something done. And I ended up going there, spending 5 years, never went back into the hospital, but things went well. And I found myself competitive for, what would be called traditional basic science tenure track faculty positions. So I ended up moving to Rockefeller, which, serendipitously was the place that Darryl Liu and Bob Heinrichsen always talked about when I was working in their labs before. And so that was kind of an amazing career turn. And then I got to Rockefeller and started my lab. It's a fantastic place to be. And I just pushed forward ever since.
It is. And let me tell you how small the world is. I actually met John Potts maybe 2 years ago, and we were talking, and I mentioned, I was in your lab, and he said, "Oh, of course, Tom. Of course, I know Tom." and I thought, it's just a small world in science.
And even another story about small world related to John Potts. I hadn't seen him for probably more than 10 years, and then 3 years ago, the year before the pandemic started, I was in Florence for a conference, and I came down from my hotel to have an early breakfast, and John Potts is sitting in the hotel dining room in Florence, Italy. I hadn't seen him for more than 10 years, and we had the greatest conversation there, getting caught up. What a great guy he is. Awesome. It is a small world.
It is. So tell us more about your first encounter with what we now know as GPCRs, and why did you continue working on Rhodopsin, and how that evolved into other now known GPCRs?
Yeah. So rhodopsin's a fascinating story to work on, and I like spectroscopy, partly because of my previous training, but partly because of my early exposure to rhodopsin. And we characterized rhodopsin function by regenerating the opsin-APRO protein with 11-cis-retinal, which is the chromophore, and then doing spectral studies on the rhodopsin. And of course, rhodopsin is a special case GPCR because the chromophore is covalently bound, but the chromophore functions pharmacologically as an inverse agonist. So once you start getting your head around that fact, there's a lot of interesting connections that you can make between rhodopsin and other GPCRs. But initially, I was focused totally on rhodopsin, and I became fascinated by the question of spectral tuning. And this question is basically revolves around the fact that all visual pigments in nature, essentially all, use the same chromophore, 11-cis-retinal, or essentially the same. There are a couple of variations in fish or insects, but every living visual system uses 11-cis-retinal. Now, then you realize that vision is tetrachromatic, meaning that some animals have more than one visual pigment that absorbs at a specific wavelength so that you can perceive wavelength-dependent information. But this visual system extends from the UV to the far red. Humans have three pigments, blue, red, and green. They all use 11-cis-retinal, but insects have UV, some small mammals have UV, birds have, a wide-ranging visual system. They all use the same chromophore. So it's pretty obvious that the origin of that is explained by amino acid side chain interactions with the chromophore in the folded 7-TM rhodopsins, or iodopsins, which are the cone pigments. So the question is, immediately you can say, wow, I can learn something about this just by making mutations in the chromophore binding pocket and see what the effect is on the spectrum. So that was a first stab at learning some useful information about spectral tuning and indirectly, about what ligand binding pockets look like in GPCRs.
Yeah.
And of course, to really take this to the next level, we needed something other than just a UV visible absorption spectrum of a mutant. So I started developing micro and nanoscale analytical methods in the early 90s. I worked with a great physical chemist, Rich Matthews at Berkeley, to develop a resonance Raman system, and had a great collaboration with Rich over about 15 years. I worked with the group in Germany, Fritz Siebert, on a microscale FTIR method so we could get IR spectra of mutants. And between the IR and the Raman, which are complementary vibrational spectroscopy methods, we really were able to come up with the correct physical chemical explanation for how ultimately a gene sequence determines the UV-vis spectrum of a visual pigment. And that all of that work extended, it sort of started in Gobin's lab, but extended for at least another 10 years, involved cloning other visual pigments like UV pigments that we did work on. It involved the work of Belinda Chang, the fantastic evolutionary biologist who's now at Toronto, that involved ancestral reconstruction of extinct pigment genes. There's so many interesting things in the visual system. And we still dabble with rhodopsin, I would say, and use it as a control, but still some interesting questions to address. But then and I would say in the early 90s, we started thinking about whether we could use rhodopsin as a model for other receptors. And that came in in a computational approach. A postdoc named Steve Lin was really good at computational chemistry. And there were some early projection maps of rhodopsin from 2D cryo-electron microscopy, not 3D, but it was really a 2D process. Gebhard Schertler and Richard Henderson and others were doing that, and they produced these projection models. So the first question was, what could you learn from that? Secondly, did that projection model apply to other 7TM receptors? So we made a pretty good model of rhodopsin, a three-dimensional model that we could test with mutants, because we could test with mutagenesis because we had made so many mutants and had such good spectroscopic methods. And then what happened was it was again serendipity, because around 1996, I started getting calls from immunologists saying, there's this interesting observation in the HIV field that the so-called co-receptor for HIV-1 is a chemokine receptor called CXCR4. It was then called fusin. It was an orphan receptor. Ed Berger at NIH figured that out. And then somebody else said, but CCR5 also is probably the primary secondary
co-receptor.
And we know a little bit about it, and so I got so many calls from people saying, we don't know what these things are, immunologists, can you help us? And so we started collaborating with a couple of people and with some companies at that time, early collaborations with industry. And we made models of CXCR4 and CCR5 based on our knowledge of rhodopsin. And we were able to transition really from rhodopsin to other traditional family A GPCRs through that mechanism. And we still work on chemokine receptors. And, you did great work on CXCR3 recently. And then the other sort of thing that happened in parallel here at Rockefeller was that I started working with Bruce Merrifield, the late Bruce Merrifield, who invented solid phase peptide synthesis. One of the peptides that he had synthesized earlier was glucagon. And so he was really the only lab at Rockefeller that was set up to do GPCR assays in the traditional sense, the old cyclic AMP stimulation assays that you know about. And so we set out to clone the glucagon receptor, which hadn't been cloned. We got scooped on that, but we quickly made a synthetic glucagon receptor and then went forward with family B studies. So we had sort of family A, mainly with chemokine receptors, and then family B, mainly with glucagon. And all of those things just lead where they lead. And
It's important to say also that taking the rhodopsin and the information that you got from studying rhodopsin and applying it to other class A rhodopsin-like GPCRs is something that, if I remember correctly, wasn't done as often. And the structure of rhodopsin came out in 2000. So, and we're talking before any structural data was published, was known about rhodopsin.
Yeah, well, we were using these rudimentary models that were informed by mutagenesis. We really use those models to try to design experiments and create hypotheses before there was a crystal structure. The 2000 crystal structure of rhodopsin, which was from native disc membrane rhodopsin from cows by Chris Palczewski and others, was revolutionary in the entire field, and really changed things, because then you could actually do what we were doing before, only now have the actual crystal structure as your starting point instead of this projection model.
Exactly.
And that allowed us to finish up a lot of the stuff we started. Remarkably, that so many things about that crystal structure were, I would say, were predicted. it was great to see the structure. There weren't that many surprises, frankly. There were some surprises about how the extracellular loop folds back in and creates like a cap on the ligand binding pocket. A few other things that were very cool. But we had essentially been able to identify all of the residues in the binding pocket already, and we sort of knew how things fit together and sort of knew the orientation of the ligand in the binding pocket. But it was just fantastic to have that structure. I guess the most surprising thing was that it took 7 years later until the beta receptor structure was solved.
I was an undergrad when the Rhodopsin structure was solved, and I spent an entire summer working in a lab, and then I think maybe a year later I got to go to a Great Lakes GPCR retreat, and I remember Chris Palchevsky being invited and giving his talk and showing the structure. And not young and naive as I was, I didn't understand the importance of that first structure. But yes, you're right. It took 7 other years to get another structure, and now I feel like there are structures coming out every week.
And of course the beta receptor structure, the helices lined up very nicely with the Rhodopsin structure, and that led to an explosion of structures. And then there, of course, was another lag. You might call it a lag, or just the normal pace of work in the technology, until the structure of the complex, Brian Kobilka's famous structure of the complex with the G protein came out. And then a lot of these things are technology driven. Of course, now there are great technologies for cryo-EM and for the imaging reconstruction technologies and the cryo-EM technologies, and the reconstitution in nanodisks, NABs, SMALPs, all the different technologies.
Yeah.
I will say that, another thing that I'm pretty proud of that started from Rhodopsin and really is carried through is the strategies to solubilize the receptors from the membranes for structural studies. We had to solubilize, most people were using cells or membrane preps from cells to do their pharmacological assays, the second messenger assays. Yeah. Bob Lefkowitz and a few other people had solubilized their receptors. That's some of his very early work, to show that solubilized receptors bound the ligand and stuff like that. But we actually had to work out details to solubilize the mutant rhodopsins. Mm-hmm. And one of the things that I did with Roland Franke was to use dodecyl maltoside. I think we were the first ones I know that dodecyl maltoside was out there and was being used for cytochromes and other things. But I think we were one of the first to use dodecyl maltoside for GPCRs. It turned out to be the perfect detergent for GPCRs, and essentially every single purification now uses dodecyl maltoside. I remember when we got the first samples from Anatrace. I guess everyone probably has heard of Anatrace, the company. We were communicating with Anatrace when it was like three guys in a garage in Cleveland, and we got a sample of dodecyl maltoside that we had to purify. So we use these big flash columns, and then we realized that the dodecyl maltoside had alpha and beta anomers that were contaminating each other.
Yep.
So we figured out how to purify those anomers, and we characterized rhodopsin function in the alpha and the beta. And they were the same, so we never published it. But we spent so much time working out the details of using the beta D dodecyl maltoside as a detergent, which is still widely used today.
It's so interesting because you mentioned the detergent, and I remember while I was in the lab, we were taking turns at making stocks of DDM, and it's a pain to make the stock. But I think it was a rite of passage for everyone who worked in the lab that they had to at least once in those couple of years to make stocks of the detergent.
Yeah. Well, of course, it's a lot cheaper if you buy it in bulk and
yes.
make the stocks yourself and aliquot everything.
Definitely. So you're mentioning the detergent, but one thing that I always found really interesting that there was available in the lab and that was developed in the lab is the not only purifying the receptor but also the nanobodies, not the nanobodies, the term
nanodisks.
Yes, the nanodisks and the technology behind that. How did you come up with the idea of enclosing the receptors in those nanodisks and characterizing purified receptors? And I'm thinking specifically at the work done by Carlos Rico.
Okay. Well, there are two parts to that, and I'll correct this. The nanodisk technology was really invented by Steve Sligar at University of Illinois. And that technology is based on the apolipoprotein in serum that transports phospholipids. So Steve Sligar sort of reduced that to practice, and we were one of the groups who was trying to get GPCRs into nanodisks early on. And there were a couple of groups that did it. Brian Kobilka was using it. Dan Oprine was using it. We decided actually to not use the human version of the apolipoprotein, but what we did was, once we figured out how the nanodisc self-assembled, we reasoned that we should be able to make a better version of this so-called scaffold protein, which is the monomeric version, just purified from E. coli or whatever. And so we screened all of the genomic literature to find the perfect apolipoprotein, and it turned out that based on our analysis, we thought the zebrafish version of this was better. So it hadn't been cloned. So Carlos, a student in the lab, Carlos Rico, cloned out the zebrafish, and then we made a version of nanodiscs that we called NABs that were based on the zebrafish version. It works great, and this technology is fantastic, and a lot of people are using it. People are doing structures in nanodiscs or NABs of GPCRs and GPCR G protein complexes. So that's a nice story. But the purification that most people are using, these affinity purifications, that was really a great innovation that Dan Oprine deserves full credit for. In Gobin's lab, Dan was able to work out the first immunoaffinity purification of Rhodopsin, and he used a monoclonal antibody called 1D4, which had been invented by Bob Mulday, who's at Vancouver. He just made a bunch of monoclonal antibodies against Rhodopsin. One of them happened to have an epitope that was the C-terminal tail of the receptor. And Dan reasoned that he could put the antibody on beads, pull the Rhodopsin out of, in detergent micelles onto the beads, wash them, and then elute the receptor from the beads using a peptide that corresponded to the epitope, which should be possible because it's a monoclonal. Yep. So Dan worked it out, and it worked beautifully. And many people use 1D4. It's a perfect antibody for immunoaffinity purification. The epitope is known. It works on the C-terminal tail with a free carboxyl terminal amino acid. And people use 1D4 without knowing its history, without knowing what went into it, and that it was used really for Rhodopsin before it was used for the other 300 GPCRs that it's been used for.
I actually knew that because of my time in the lab, and I've aliquoted some of the 1D4 antibodies as well back in the day.
Yeah, it's a great method, and Dan deserves a lot of credit. Everyone who uses that strategy should really cite that early work. But as usual, people forget and they just pass it along. But it's a great strategy. It works better than FLAG. It works better than HA. It works better than all the others.
Agreed.
Yeah.
And then the other question I had for you. So we've talked a little bit about what type of projects you started working on, moving from MIT to Rockefeller, and then working on chemokine receptors as well. And then you moved on to more computational work with some of the people who were in the lab and the RAMP work specifically. Can you tell us how you got into working on RAMPs?
Yeah, the RAMP story. Well, the RAMP story goes back to the family B interest, really working on glucagon receptor and GLP-1, and then following the literature and seeing these amazing accessory proteins. That RAMP isn't the only accessory protein, but it's kind of an interesting one because it's a single spanner that has this sort of chaperone function, gets receptors to the surface. Steve Ford, one of the great papers in the GPCR literature, the history of GPCRs, the McClatchy Nature paper with Steve Ford. So it has chaperone function. It explains a lot of the weird biology of GPCRs, how you can express a receptor in one cell and then express it in another type of cell, and it has apparently totally different functions. Could be because of RAMPs. And then it turns out that RAMPs also affect pharmacology, and also in the family B story, those papers are amazing. And even now we see the recent very important monoclonal antibody drugs against receptor RAMP complexes that are used to treat migraine. And it's a great story. So we've been trying to get back to that. But fundamentally, we've always been interested in protein-protein interactions in bilayers and what drove it. We started to approach that both computationally and experimentally with Thomas Huber in the lab, working with Michael Brown on some cool experiments showing that there are some physical properties of the membrane that affect whether receptors aggregate. Then we started being curious about whether receptors that form heterodimers, two different receptors forming a dimer unit, whether there are protein determinants. There must be. What are they? And we started, again, applying this to the chemokine receptor question. And very interesting and important heterodimer in that field is the CXCR4, CXCR7 that you contribute a lot to. And then we, through Thomas Huber, we collaborated with Jan Siewert Merrick, who's the guy that invented this fantastic strategy or contributed to it called coarse graining, where you could do relatively large, complex systems in membranes, say a reasonable membrane model and 12 receptors, and simulate what happens. And this coarse graining allowed us to do some really cool work using mainly Rhodopsin again on figuring out what the principles are of self-association of receptors and bilayers. And that work is sort of still ongoing, or we're still interested in that subject. But we always have this tension, I wouldn't say tension, but collaboration in the lab between the computational and experimental approaches.
Yeah.
And And of course, there's so much that can be done nowadays with BRET strategies to look at protein-protein interactions. Proteomics methods, although they're challenging with membrane proteins, are really coming online. You see work of Carol Robinson and others with mass spec of essentially native proteins. And then you see proteomics methods like we're using with these suspension bead array systems where you can interrogate hundreds of potential interactions all at the same time. So I think that we're at a point now where technology is driving a lot of the advancement and also miniaturization. So you don't need to have a lot of material anymore, even to do structural studies.
Yeah, I think it's really the missing piece when it comes to interrogating receptors and receptor function, but also receptor conformation and figuring out how many receptors are there in a complex and how that complex drives function. Being able to run assays in plates. And I say this oftentimes, and I think Brian Roth and I were having this discussion during our podcast, is the fact that he says he loves BRET. I love BRET too. And being able to having a robot and having a plate reader and just running plates and acquiring massive amounts of data, and then having the computational component there to go through and analyze the data is what was missing. And I think now it kind of came together.
Yeah, of course, BRET is fantastic. Many people love BRET. BRET doesn't always love those people because it takes often a while to get it to work, get it up and running, getting the ratios correct, all that. But the new, much brighter reagents that are available for those systems have really made it accessible to everyone. And Brian's contributions have been fantastic. Michel Bouvier, fantastic contributions, and a lot of other people. But it's really a transformative methodology.
I'm biased towards it. I think it's just an easy way to go about it. But I have to admit, depending on which system you're looking at, on which receptor, sometimes it can be more of a headache than anything else.
Well,
it's science. I think that's the price to pay for
everything's a challenge, but the goal is to create as much data as possible. So
exactly.
That's one way. BRET is certainly a miniaturizable method that is very powerful to look at signaling. And the other key point that's come out of work in the last decade from Bob Lefkowitz and others, of course, Bob and arrestin signaling has been a great advance. We knew about arrestin signaling in the visual system before, and that's another thing that's analogous in terms of what's happening at the receptor. There are a lot of similarities there. And so arrestin signaling is important, but the concept of signaling bias is really what's cool. But it does mean that you can't just rely on any one single assay, because you have to consider G protein signaling and arrestin signaling and, who knows what. So you have to consider more than one pathway, but you can't just assay multiple different pathways in multiple different cell systems. You have to be able to really compare in similar backgrounds. So it's important. And we've spent a lot of time in the last couple of years working out strategies to be able to do expression level, cell surface expression, G protein, second messenger assays, and arrestin recruitment assays all in the same cell under miniaturized conditions. And we've been interested in that because we've been working on a receptor that is constitutively activated in pathological states. And it's hard enough to do ligand-activated signaling where you have a good dynamic range to work with. But if you're trying to measure signaling bias in constitutively activated receptors, it's even a bigger challenge because the dynamic range is much reduced. So that's what we've been focusing on.
May I ask which receptor system are we talking about?
Oh, well,
I think I know the answer to that, but I think I know
we got interested in this receptor called cis-LTR2. There's a story, sort of an interesting story behind it, but it's basically it's long been known that uveal melanoma, which is an eye cancer, common eye cancer that's from the back of the eye, from the choroid, the pigmented part of the eye, or can arise in the iris, which is the anterior pigmented part of the eye. That this tumor is a melanoma, but it has a very low mutational burden, unlike cutaneous melanoma. Yep. And the defective signaling pathway in uveal melanoma is always the GQG11 pathway. Most of the mutations in that are single mutations in GNAQ or GNA11 that activate the alpha subunit of the G protein. So this was kind of a very interesting biology. But then we started working with collaborators at Sloan Kettering to see if the G protein is activated, is it possible you could find upstream GPCRs that couple to those that could be also activated and cause the same phenotype? Okay. And we did. We found cis-LTR2 in about, not many, maybe 3 or 4% of all patients with uveal melanoma. They have a mutually exclusive mutation, meaning that these patients have an activating mutation in the receptor and not an activating mutation in the G protein, and the phenotype is essentially the same. And this receptor, cis-LTR2, is a cysteinoleucotriene receptor, and it has a very interesting mutation that we had noticed earlier in some of our work on Rhodopsin, and it's L129Q in the third transmembrane helix, and it basically pries open the helices, affects the NPXXY, and causes the receptor to be activated. So that's cool, but then you say to yourself, as we've just been talking about, well, if the receptor is activated and it's on the cell surface and it's signaling through the G protein, why doesn't it also recruit arrestin and get desensitized and internalized and turned off? Well, that's the interesting thing, is that the mutation causes really a profound signaling bias. The mutation activates the G protein signaling, but doesn't recruit arrestin. So it's a fascinating story, and it's a bona fide oncogene, and one of the first GPCR oncogenes. So we're very interested in this receptor. It turns out that 8% of all human tumors in databases have mutations in cis-LTR2. It's like the second most mutated GPCR in any tumor databases. And so we're interested in devising ways to screen tumors for activated GPCRs. The idea being that GPCRs are, druggable. We're all in this field in part because GPCRs are druggable. Shouldn't we be able to find drugs to turn off aberrantly activated receptors in cancer? Should be possible. So that's what we're interested in right now, in addition to just learning about the pharmacology of this really fascinating receptor.
It's interesting because last year, I think the second or the third episode of the podcast was with Sylvia Goodkin, who also is interested in uveal melanoma, but from the GQG11 perspective. Mm-hmm. And I thought it was, and I was in the lab when you guys started working on cis-LTR2, and I kind of knew the story, but now we have both pieces together. So now we can understand what are the two main components and driving components and mutually exclusive components of uveal melanoma.
One of the great things about your podcast idea is that you talk to different people and are telling the stories, and you can see how all of these stories and the the story arcs of GPCR research interact. We haven't collaborated with Silvio, but we read everything now he does. We're interested in his work, and it's brought a whole new flavor to the lab to work on something new.
Yeah, and interestingly, we were talking about the ramp story. I spoke to actually Steve Ford this week, as we recorded a podcast episode this week, and we were talking about the story of the ramps and the migraine model. And he had mentioned that once they had cloned the ramps, they shared the materials with a bunch of scientists in academia, and everyone he mentioned I've already spoken to. Name, for example, Debbie Hay from New Zealand. So it is really a small world.
Yeah. It's true. And, the thing is that science is really, I think the general public doesn't understand science. We can see that in the age of COVID, where the public is having trouble figuring out who to trust and who's an expert and who's not. And I think the public has this idea that science is an impersonal type of a career. But it turns out that I don't find it to be that way at all. I mean, I organize my thinking about people in the field, not subjects necessarily, because in general, the best people in the field work on a lot of different things at the same time. They may be thematically related, but people are going to move around. The best people follow their interests, and so you want to know what people are working on, and people have to communicate. We probably don't communicate well enough to the general public. We're used to teaching, arcane subject matter and very technical things, but we should be able to explain what we do. We should be able to explain to an average person who can read the newspaper, why it's important to take a COVID vaccine and what the science is behind it. It's not that complicated.
Yeah, it's not. And I think I've seen on LinkedIn, I think there were a couple of images where someone took actually the time to make a table or figures and explain the whole process and the differences between the different vaccines developed by the different companies. And I thought it was such a great thing to have out there, and just, invite everyone to take a look and better understand science and the science behind the vaccines. It was such an ominous moment when the needle went into my arm, and I thought to myself, I feel like it was just a fraction of a second, and it was in, but then all the work that went into getting to that point is just unimaginable.
Yeah. I think there's some information gap. Of course, information is out there, but you can't expect the average person to read the papers and try to parse everything. And there's probably also a belief gap. Some people just don't believe that it's the right thing to do. So somehow information and beliefs have to be respected, but also we have to tell people what the facts are about these vaccines.
I think one of the issues is that there was too much information out there, and it's not properly curated sometimes, and you can read many things, and if you don't have the scientific background, or you don't know how accurate that information is, then you cannot decide as a layperson, does this make sense or not?
Mm-hmm. Well, I think people are also seeing science happening in real time, which is not usually the way it happens. And that's disconcerting. It's not a completely smooth linear pathway. There's a lot of ups and downs, as everyone who works in the lab knows about the ups and downs. And sometimes experiments don't work. It doesn't mean that the theory's wrong or that molecular biology doesn't work. It just means that your experiment didn't work for whatever reason. You have to keep going back to it and figuring out what the underlying issues are.
I always took it as a sign when if it was Friday and the experiment failed, that was the time to just close up everything and just go home and come back the week after. Sometimes it's just not meant to be.
it's always better to work harder when things are working and not when they're not working.
Exactly. I think what I learned during those 5 years of the lab is in your lab is making the difference between working hard and working smart, and then trying to always work smart and not hard. But sometimes when things are working well, when an experiment is working, don't stop, just go for it and acquire the data before anything happens. Mm-hmm. And if there's any misalignment of the stars that make the assay stop working.
Well, I have this sort of affinity for a results-only work environment, as I say. I don't really take attendance or push people to be in a certain place at a certain time, but eventually it's nice to get some results.
It is, which is one of the reasons why I really loved being in the lab, because if I needed help, you were there. If I didn't need help and I could roll on my own, I was free to roam the hallways anytime I wanted to, and had access to all the instruments I needed, and the proximity. I think that's one of the best things at Rockefeller, is that I used to live 5 minutes door to door from the lab, which was a blessing and a curse at the same time. But going into the lab on Sunday night for 5 minutes, and being able also to walk the dog while going to the lab, who was a lab member. There were moments when he was very happy of me walking him, and then when he realized that actually we're going in to do some work, then he wasn't so happy about it afterwards. But it was still a luxury that I'm very happy to have been part of.
I had a conversation with another former postdoc recently, who was in the lab before you. You wouldn't have known who this person is, and I'm not going to blow her cover. But she said, by the way, did you know where I was most of the time when I was a postdoc in your lab? I said, and she was one of the most productive people ever to come through the lab. And I said, not really. I'm not sure what you mean. And she said, well, I became obsessed with New York and obsessed with, independent films. And I used to go to these film festivals like for 8 hours a day, 5 days a week. And I would come into the lab at 6 p.m. I would be in the movie theater all day and come into the lab at 6, and I'd hardly ever see you, but somehow it didn't matter. Still got the work done. And she learned a lot about avant-garde culture.
That's amazing. 2 more questions, Tom. One is, well, I think we know the answer to that, but what's your favorite GPCR?
My favorite GPCR is Rhodopsin. I have a lot of GPCRs on my hit parade, but that's got to be number one.
And then the other question I had for you is, if you had to give advice to young scientists, junior scientists who want to contribute to the field, what would that be?
Stay connected. Use these resources that I never have gotten very good at, but stay connected, and really find something that you're comfortable with and good at, and become an expert at that. Learn everything you can about whatever that thing is, and then talk about it often, frequently, as much as possible. Promote your own work. Be a little bit more of a self-promoter than you think you should be, but in a nice way. And then reserve some time to do other things that are relevant and related. That's not very good advice, but
well, I think it is, because if you're a PhD or you're a scientist, you have to have a specialization. You have to know your thing, but for that you have to explore and figure out what is it that excites you and what you love, and then if you want to be the expert, you just have to deep dive into that.
Yeah, you do have to be recognizable for something, and hopefully it's the thing that you're most excited about working on. You don't want to be recognized for something that your former boss was interested in, or that your thesis advisor was interested in. You have to have some level of independence and make a mark for yourself.
And then from that, you touch on a very important point. How do you, and maybe it's a tough question, how do you become independent? How do you figure out what question you should be asking to differentiate yourself from your former group leader, and at the same time be successful? Because you mentioned going into Gobin Khorana's lab and say, "Oh, I think 2 years should be it," and Gobin saying, "Well, no,
Yeah.
You need more than that."
Yeah, well, this is an important question. It depends on your career track and your aspirations, whether you're interested in academics or in, more in industry or commercial, whether it's biotech and entrepreneurial stuff or big pharma, for example. All those things have slightly different requirements. But I remember talking to Gobin when I was leaving the lab. Of course, I knew I was heading to an academic career, and it's hard to become independent of a Nobel laureate with 20 postdocs who can work on anything they want. So I was thinking maybe I should talk to him and have that conversation about what I should do now. And so I did. I went and said, Gobin, I was thinking maybe we could agree on something that I could work on for a while, that I could work on myself, and maybe you wouldn't want to work on that. I have some ideas. He said, no, absolutely not. I'm not going to have this conversation with you. How about this? You can work on anything you want to work on, and I'll work on anything I want to work on. And so I was like petrified that whatever I worked on, he would be working on. Anyway, somehow there was divergence along the way, at least enough divergence that people thought that I could work independently. But it's a fraught question. I think nowadays it might be a little bit easier because in the old days, everyone was doing the same basic technique. Now there are many different strategies. You could become an animal model of disease expert. You could become, MST expert, a bread expert. You could really become an expert at some technical area. You could be a cryo-EM expert, whatever. That was harder to do in the past. So you have to be an expert at something to succeed. Agreed.
With this, I want to thank you for your time, Tom. It was a great pleasure talking to you, and I will talk to you soon.
Thank you. Best of luck.
Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, Alexa Druan, and Ines Pinero. We look forward to seeing you live at the Dr. GPCR Summit in September. Don't forget to visit drgpcr.com/summit. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us testimonials at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.